Yeah, the first was the comparison actually of de novo sequencing and the realignment. So we did the de novo sequencing, so it's probably there for you. If not, then you can just redo the de novo. This is not de novo assembly, actually. This is a realignment, but filter out non-aligning grids and okay. So yeah, this is when you do a realignment. So when we did the realignment, we didn't do actually the filtering. So if you do with bow tie, that's one of the options. One is use bow tie. 2 and use the minus listing minus un conc. Let me just put this here right next to this one. So put the non-aligning reads into the file, into a separate file, or the much easier option is to use snipping that will take the reference sequence, will take your short reads, and with the snippy you have to use the minus unmapped option, and again minus unmapped and the file name. Okay, that's the first step. Of course, if you want to check the assembled context for virulence and resistance genes, this is what we did with the abricate. So you will have a list on that, and again the same thing goes for the. So yeah, actually, if you use the abricate, I think we missed this thing that it doesn't do all of these predictions. It only uses the NCBI database, and I'm not sure that the NCBI database is involving the virulence genes as well. So if you say abricate minus list, then you can see that you have the NCBI database, which is the default database it's using. I'm pretty sure that all the antimicrobial resistance genes are here, but I'm not sure that the virulence genes are there as well. So if you want to check for plasmid sequences, then you have to say minus setup, no, minus db. So abricate. If I'm just quickly show you, if I go to my folder, and we have the context FOSTA here, which is one of our FOSTA files from, I think, from de novo assembly, and we say abricate minus db plasmid finder, and then the FOSTA file. Then it will use the plasmid finder database, and it will look for plasmids. And these are now plasmid identifiers, rep 19, 16, 7a, and so on. And actually with very high coverage. If you want to look for virulence genes, then I think it's the VFTB probably. So do the same thing. But again, it's not the same what you will get back. So it's not doing, I don't know if you can ask to do all databases. I don't think so. I don't see that kind of option. So VFTB, which is the virulence database, I hope. Maybe there won't be any. There are actually quite a lot of genes. Okay, so here again, okay, so this is the same, and of course the difference is that here you are running the abricate on the de novo assembly, what we did before, and here you are running the abricate on the reference genome, because in default, if we just do the bowtie 2, you don't have the FASTA file for your own sequence. You just have the FASTA file for the reference genome, and you have the BEM file. So there is a chance that you don't have something in your sequence which is present in the reference genome. So you will annotate genes that are not really present in your sequence. And there's also a chance that there are extra sequences in your sequencing, but you will not annotate or not found, or not, will not find, because it wasn't aligned to the reference sequence. So that's why there is this extra line, and this is not working. So please don't use this. But we saw that if you use snipping, then it will also create a consensus file. It is called SNP's consensus.fa in default if you don't give any other name for it. So as soon as you have this file, you are just doing the same kind of abricate on this file, and you will see what was assembled from the genome. And what you have to do is just put the two lists next to each other. Look what you saw in the de novo assembly, and look what you saw in look what you see in the realigned FASTA file. And probably you will have quite a lot of differences. I haven't checked it, but if anyone has the list, please provide it to me. I'm pretty sure that there are many genes that are not present in the reference genome, but will be there in this sequencing file. And the second one, I don't know if anyone did the second one and has any idea what is the other species, what are the two species that are mixed together. So at least the first step was done. No de novo assembly. Okay, I won't give you the answer, so you have to find out. But of course, the first step is just a simple de novo assembly, as we did. Space pipeline, or you can use the shovel as well. That's the other option, which will give you at the end the FASTA file for you. Oh, actually, this is going after this point. So this is what you do to gain the FASTA file, and then use some kind of program. We used mesh, but you can also use checkm, use mesh or checkm to find out the composition, species composition of the And then you can use again bow tie with the same kind of minus minus un conc. So you will have two species names. You have to download reference sequences for both CBI, for example. And then, as we spoke, if you are more interested in one or the other, then always use the less interesting species. So you use the less interesting species to align all the reads and extract the non-aligning reads, and then assemble the non-aligning reads. But in this case, I think both species will be very interesting. So if you want to do in both ways, then extract or use both reference sequences to extract the non-aligning reads. Of course, in this case, you may think about how can you assign the novel genetic element. So just imagine that you have two bacterial species mixed together because you accidentally picked up a colony, which wasn't a monoclonal colony. You assemble the genomes, you find out that there are two different species there, and you, of course, they are, if you are lucky, they are both well known, so you will have the good reference sequence in the NCBI. You download it, you do the realignment and do the separation of reads. But even if you do the separation of reads, maybe you will realize that there is a bunch of reads that are not aligning to any of these reference sequences. For example, because one of these bacterial species are, one of them is containing a plasmid or more than one plasmid, or maybe both of them are containing plasmids that are not involved or not present in the reference sequence. In this case, as far as I understand, it's very hard to decide where to put these plasmids. Of course, if you then assemble those plasmids and you characterize them, and one of the species is an E. coli and the other one is a Staph aureus, and you find a plasmid that was always found in E. coli, then there is a high chance that it was actually in the E. coli. But the problem is that when you are picking up a colony that looks like a monoclonal colony, as far as I understand, but please, Ibrahim or the other microbiologists, just correct me. So usually, if you pick up something that you see as a single colony, probably, but it's a mixed one. It means that the bugs are looking quite similar. They are liking the same kind of culturing conditions, which potentially means that they are not too far from each other. I don't know if this is true or not, but I can imagine. I guess you cannot really grow Staph aureus and E. coli just right next to each other that easily. So in this case, if they are close to each other, then there is a chance that their extrachromosomal DNA is also compatible with each other. So even if you find a plasmid that was found in one of your species, but your other species is very close to that, you cannot really decide, I think, with 100% reliability where the plasmid were. So probably the best thing is going back to the sample and try to reculture the species separately, especially if there is something very interesting, you know. If there is nothing on that plasmid which is interesting, then don't do it. But if there is something very serious there, an antibiotic resistance gene that is important, then you have to go back and be absolutely sure that it is in species A or species B. Am I right, Ibrahim? What do you think about this?
Yeah, more or less. I mean, the main problem is people don't do when they are isolating strains, so they don't do the purification step properly. So if they just pick a clone from the original culture plate, it's quite likely clones grow on top of each other.
Yeah.
So I mean, you see, although it is well isolated, it might be mixed still. So that's why you normally like do another step of purification onto a new plate from a single colony of the original plate, and then you normally will have a much quite guaranteed to get a quite well separated colony. That's why how you minimize the chance of contamination. But again, contamination happens sometimes as well. So.
And if you do the second so-called purification, so yeah, just spread again one colony. Do you only pick up one colony at the end, or
one colony? Yeah, and then you
always pick up only one colony.
Yeah, and then you because when you culturing for DNA extraction, you just pick one colony from a plate.
Okay. Okay.
So that's why some people are working on multiple colony sequencing techniques or strategies to see the diversity of the certain species in the host.
Yeah, I think it's okay to do, but again, I think the main problem is when you are facing with those kind of genetic material which is not known, which is new or which is extra chromosomal, and we will never be able to. It's the same as we do with metagenomics. Of course, there is way much more diversity there. Okay, thank you. So here, yeah, it's extracting the non-aligning grids. I just copy this one here because it's the same way how you can do this. And this is actually, and again, a de novo assembly, which is the same as this one. And if you want, you can also check all of these. So, okay, I will just have this, put this on the Google Drive. So if you want to do this, there will be some help how to do it. Right, but this is not the topic for today. So, okay, for 20 minutes. So what we will do today is, and you can all log into the server because we will do quite a lot of things, and everything is very fast and everything is also quite easy, I think. Is there anyone who wants to be my helper today? Or it is my duty to pick someone? Tom? Tom?
Yeah, me.
Okay, are you okay to help?
I can give it a go.
Actually, I'm not sure that there will be quite a lot of need for help. So because what I will do mostly is you can all do, and you all have to do. But if there will be any kind of installation, I think there won't be. But if there will be, then I will ask you.
Okay.
Okay. But I think most of the things are already there. So probably just follow me. Everyone just follow me and do it together. Okay. So what we will learn today is one of the, I think, the most basic thing you will do, and one of the things you will do the most of the time if you do any kind of bioinformatics. Of course, if you are only looking for the whole genome sequences of your hundreds of isolates and don't want to go into the details, you just want to use those specialized programs we already discussed, then it's not that important for you. But as soon as you want to go into a bit more detail, if you want to find a sequence, if you want to find a gene, a domain, or anything like that, and you don't want to do this manually one by one on a website, So, for example, if you don't want to do BLAST on the BLAST website, because you have, again, hundreds of sequences, then this is the way how you will do this. I don't say that I don't use BLAST on the website. There are so many cases when I have, for example, one protein or one gene or one DNA fragment. I would like to find out what's the function, what does it look like, what's the homologs for that sequence, and then I just simply go up to the BLAST website and insert and wait until the BLAST website finishes. But in many cases, you have more than just one sequence, and you want to do look for your genes of interest in more than just one sequence. So we will discuss 3 approaches, and I hope that we will be able to discuss all of them today. It's actually 3.5. You will see why I'm saying this. The first one is BLAST. Of course, probably all of you already know what the BLAST is. So you have a sequence, there is a database, and you want to find out if your sequence is presented in the database or not. The other way will be looking for sequences, or most likely looking for domains by using a hidden Markov model-based search. And the third one will be looking for exact known sequence patterns, even if it's a DNA or a protein sequence pattern. So the BLAST is, I think I sent you a link about how to use BLAST, but yeah, the documentation was here somewhere. There is a very long user manual if you want to read it. It's really very extensive. But what you have to do is, actually, there are 4 programs you may use, and there is one extra. Luckily, all of them are already installed on the bioinformatics server. I don't know which program it was maybe the Proca or the Snippy, or I don't know which one. But if you just say BLAST and then push tab twice, then you can already see that we have the BLAST N and BLAST P and BLAST X. And we also have I think the 4th one is the N and BLAST X. BLAST N, BLAST X, or P BLAST X. There's a 4th one. So, okay, let's just go through them and we will see. So if you say BLAST N, that's what probably most of our geneticists will use. So the BLAST N is -H. This is the program if you want to align nucleotide sequence to nucleotide sequence, nucleotide BLAST. So you have a nucleotide database and you have a nucleotide query sequence. The brother of BLAST N is BLAST P, which is the protein BLAST. So you have protein database and you have protein sequence you look for. And then there is the BLAST X, when you have the translated query. So this is, in this case, the query is DNA sequence, I think, and the database is protein. I didn't use these ones too often, but I'm sorry, I just usually use the 2 basic ones, BLAST P and BLAST N, and I always mix up the 2. But I think this is when you are aligning, when you are looking for DNA sequence in the protein database, and the BLAST X is just translating for you And the fourth one is the opposite, when you have protein sequence and you are looking in DNA database. But I think the most important thing is to understand how BLAST N and BLAST P works. Actually, just BLAST N, because BLAST P works in the same way. And there is one extra program you have to know, which is the make BLAST DB. That is the program which will do the database for you. Okay, so let's go forward. And what we will do is just simply, first we will do a database, and then we will do some searches on the database. If you go to your own folder, and I think we don't have too many sequences here, but we have something in the data folder. So we have, for example, the GCF, this GCF FNA file, which was the reference sequence for the Staph aureus. We already made a database for the bowtie2, but we cannot use, of course, the bowtie2 database for BLAST. So let's just go to your back to your own directory. And what we will do is we will use the make BLAST DB first. So this is how you use it. So you say make BLAST DB, then you are saying what's the everything which is in square brackets is not obligatory. Everything else is you have to say, and you also have to give the files. Yeah, I mean, this is important because otherwise you don't have an input file. So what we have to do, we have to say make BLAST DB minus in the input file, which is our genome file. You can say an output file here, minus out database name, and then you will define the database name. If you don't define it, it will just make the same long file name. It will use the FASTA file name as the database name. And we have to also say minus DB type and molecular type. If you say make BLAST DB minus help, then you can see that there are two different options here. Minus DB type can be nucleus or prot. Of course, we will use nucleotide because this is a DNA thing. So the first thing what we have to say, DB type. Of course, you can use the DB type first and then the minus in. It's there is no obligatory order you have to follow. So this is nucleus, and then minus, just simply minus in. If you are in your own Scratch folder, then you just have to go up by 2.data/GCF and tab, and this will be the FNA file. And because we don't want to put our database there, we also give the minus out. We can give a much shorter name as the, I'm checking the chat. Okay. I don't know if you have your, yeah, so sorry, because you just logged in. Tom had a question, and so of course, when you are logging in, you are just in your home directory like this, but with no colors. Only I have colors. So you have to say cd/scratch, and I don't know if you have your, yeah, you have your folder there. So just go there. I'm going into my own folder. Okay, so you just have to change your directory. We are always working in the Scratch directory. So make BLAST DB, I deleted it, but it's not a problem. DB type nucleus minus in ./data/GCF, and the long file name minus out, which will be the output file name. Let's just give the st our plus db as the output database name, and that's all. So you are making the plus db, you are giving the type of your input, you are giving the input file, and then you are giving the output file name. Of course, you may ask, why do you need to do this? Why not just simply search a sequence within another sequence? The answer is because it's way much faster if you pre-format the database. So there will be a lot of different like indexing and other things that will make the BLAST-N program much easier and much faster to search in this BLAST-DB. So that's all. If you press enter, it is happening extremely fast. And if you look into your folder with ls-ltr again, it's very useful to memorize this command. Then you will see at the bottom your newly created files. And as you can see, there are 3 new files here that was created, and these are the files you will need for searching. I think maybe the easiest search is just to simply look for the context FOSTA. There will be quite a lot. Maybe we can just make a smaller piece from the context FOSTA. Let me just, so what you can, oh yeah, if you have the context FOSTA head there, then you can just simply say head -n. Let me just first see the context FOSTA. Yeah, the first config in the context FOSTA is 500,000 base pairs. So let's just print out the first 100 line. Actually, we don't need 100. Let's just make 50 and put this into short config piece .fa. Okay, this is again very fast. It's just print out the first 50 line, which means that it will contain the ID line and 49 lines of sequence. I think in one line there is usually, I think, 120. I'm not sure. We can always check. We see, oh, so if you say grep -v, you already know what is grep. Grep is the program if you want to find a line in your file by using pattern search. We usually do this, grep -c, this one in the short context. If you do this, it will look for lines which are starting with this sign, or which means that it is a FOSTA ID line. This thing means that it's starting with, and this is just the text we are looking for. And if you don't use -c, then it will just print out to your screen. If you use -c, it will count. And you can see that there is only one line with a FOSTA header. If you say something like this, I'm not sure that you can do this, but yeah, you can do. So the -v means that exclude the line which is starting with this sign, and then with the -c count it. So as you can see, we have 49 lines without the ID line, so 49 lines of sequence. And if you don't want to count this, but if you want to print out, then you can also print this out. If you just simply press this, it will be printed on your screen. If you want to count how many nucleotides you have, you are just simply piping this output into the next program, which is the word count. And you can see with the word count that it has 49 lines, 49 words, because of course one line is one word for the program, and about 3,000 nucleotides. So it's a nice big piece of DNA which we can look for. in our reference genome or database. Of course, this is just an example. Usually you are looking in a much bigger database, not just one genome, and you are having more than just one 3 KB fragment. Okay, the first thing is that if you just simply use BLAST_n, if you go up to the options or the usage, it's quite extensive to help. But if you go back to the very beginning of the BLAST, then you only have to say BLAST_n, and then you have to give the database name -db database name, and then -query. I don't see where is it, but -query, which will give you, where you will give the query sequence. Yeah, it's like this. So that's the basic usage of the BLAST. Let's just try out BLAST_n -db. This will be our Steph overuse database, which we just made. So this str+db. You don't have to give the extensions, only this thing. So up to here, this is what you have to give. It will know that it has to look for a .nhr, a .nin, and a .nsq file. And then -query. And this will be our short config piece.fa. This is very easy. And this is completely useless, as you will see. The problem is that, of course, if you scroll back and you see that, oh, that's very nice. The sequence was found and the config is there. There are some gaps here and there, but actually the alignment is quite good. But this is not really what you want to get back if you, for example, looking for a gene sequence in hundreds of sequences, or if you are looking for hundreds of fragments in a database, because then you will get back this text file and you have to go through the text file manually. And the whole aim by running BLAST in the command line is to make it as automatic as we can. So the first thing what we have to do is to change the output format. And I don't really know why NCBI gives this as the default output format, because again, you don't really run BLAST in command line if you want to see something like this. If you want to see something like this, just go up to the website, and there you can see in a graphical user interface, which is much better to see, much more user-friendly. If you want to do something on the command line, you want to get back something which is much easier to parse, which can be put into Excel if you want, so much more like a table. Luckily, there are quite a lot of different options for output. So again, if you say BLAST_n -help and scroll up to the output section, formatting options, and there is this -out fmt. So that's the output format. And there are so many different versions you can use. There is an XML file, which is very useful if you really want to take out all the details, but it's not really human readable. There are different text formats. There are these new type things, these JSON files, which is a kind of a new standard in data format. And also there are, I think, different kind of FASTA files or alignment files. But I think that most of the times, if I'm just thinking about my BLAST searches, in 99% of my BLAST searches are ending up in a tabular format. So this is the table what you can take and put into Excel easily. So what you only have to say, together with the previous command we did, is -out fmt 6. So if we scroll up or go up to your previous BLAST and command. The exactly same, the DB and the query is the same, but you also say -out fmt 6. So the output format should be version 6, which is a tabular format. And if you do this, then it's way much easier to see what is happening here. So what we have here is that this is the identifier of our query sequence. This is the identifier of the subject sequence, so where this sequence was found. And there are a bunch of other columns here. Unluckily, there is no header, so you don't know what is that. How can you find out what are these columns? You have to scroll up again and go to the output format section. So this one here, and give it a go down, and you can see here a lot of different field identifiers. Your extra columns will be several from this list. If you go down, this is what you can find out, that in default, when there is no list provided what you want to see, then this is what you will get back. So QA CC where, if you go and look for it, that means query accession and version. Okay, this was the first column. Second column, SACC where, this is the subject accession and version. So the query is your sequence, the subject is always what was found in the database as a homologous sequence. And then parent, next one, which is the percentage of identity, as far as I remember. Yes, parent is the percentage of identical matches. And then so on. So there is a length and mismatch, gap open, Q start, Q and S start, S and these are, I mean, this is quite important because this shows you what was the length of your alignment, what was the length of the match. I don't really use these two too often, but it can be interesting. This is where your query starts and query ends, and this is where your subject starts and subject ends. So in ideal world, if you have a query sequence, which you assume that it will be completely in your subject. So for example, you are looking for a gene, or you think you have a gene from the beginning to the end. So ideally you want to find the whole gene in your BLAST database. So ideally the Q start is 1 and the Q end is the last nucleotide. So it's the length of your query sequence. And this is also very important because this will give you the position of your sequence within the subject. So if you want to look for the position of your gene in your genome, then you will find the position of your gene within the genome here. This is also important, and also the e-value is usually very important because that will give you a kind of estimate on how good is the hit. But the e-value is depending on many things. So I much more often use the percentage of identity as my determining factor if I accept or not accept the hit. The reason why we have more than one here, so now that we are knowing what are the fields, so as you remember, we have about 3,000 nucleotides in the config. So the config was found perfectly with 100% identity and full length in the subject sequence from here to here. Okay, the e-value is 0, which means that it has a perfect match. So you are always trying to have as low e-value as you can. But it is also it was also found in other places, because probably there are some fragments within our short FASTA file that can be found in other places in the genome, sometimes with 100% identity, sometimes with a bit lower identity. But as you can see, all of these alignments are way much shorter. So these are just small pieces of our sequence aligned to different places within the genome. Okay, so, but of course, the first one is the most important one. There are many ways you can refine your search. So for example, you don't really want to look for these ones because it's not important for you. You just want to look for those hits, which, for example, at least covering half of your query sequence. So for example, you are looking for an antimicrobial resistance gene, and you want to find at least a half of it somewhere. Then you don't really care about the small alignments. You just want to see a very high identity and this one very close or not too far from your query sequence length. So in this case, you have to add an extra column. We don't have the length, the original length of this thing. I mean, the original length of our short quantic piece FASTA here. This is actually now matching with that, but it's not here exactly. So if you look at the output fields, there is an SLen, the subject sequence length. Oh no, sorry, not this one, but the QLen, which is the query sequence length. This is not involved in default in the output. QLen is not here. So what we will do now, I will copy this from here, and you may copy as well, and we will modify the output. So I just go up. It's a bit strange because the way how you have to do this is, oh, nice. It went to a new line, sorry. I didn't want to do this. Okay, I can use this. So you have to put the 6 into the quotation marks as well. It's a bit strange, I think, but it works like this. So with the out FMT, you have to open your parameters here. First, you have to put the 6, which gives which means that you will output a tabular format. You can leave there the accession of the query and the accession of the subject, of course. You have to leave there the percentage of identity because that's one of the most important things. You have to leave there the length. This is the alignment length. And it's not that important now where the hit is. We just want to see if it's there or not, okay? So just put here the QLen, and that's all. And of course, close the quotation, okay? So out FMT 6, and then these identifiers, and then the QLen. We will get back exactly the same amount of hits, of course, but with much less columns. We only had the 2 accessions, the identity, the length, and the subject length. Of course, this last column is the same because we only have one query sequence, and the query sequence length is always the same. But what we want to do now is that we only want to keep those lines where this alignment length is at least the half of this one, okay? This is not something what you can really or easily do, I think, in BLAST. There is a percentage of, I think, yeah, percent query coverage per HSP. I think maybe we can do with this one. Let me just try, but I usually do this with some kind of post-processing. Okay, that's working as well. So if you use minus cov underline HSP underline like percentage 50, that you are asking the BLAST to only output those hits that are at least covering the half of your query. And of course, then all of these are excluded. But I think, so first of all, you can just output everything and then you can process in R if you want. It's very easy to process in R. You just keep those lines where this one divided by this one, oh no, sorry, this one divided by this one is more than 0.5. But you can also do this on the command line. And let me just show you a program which is called Oak. This is also a program which is always installed in every Linux and Mac operating systems. It's a very, very useful program. It's actually a programming language, but you can do very simple tasks very easily here. Let me show you one of the most simplest one. If you just want to print out the first column here, of course, there are other programs you can use for that as well. But if you just want to print out the first column, you just simply say print $1. And then you will get back, I mean, this, of course, this is, if you look at the whole line, the chat window here. So this is the output. This is the command for the BLAST. And instead of putting the output to the screen, we are giving the output to the Oak and ask Oak to print out the first column only. There are many cases when you just want to check if your gene was there or what kind of genes were there. So just imagine that your query sequence is your whole genome and your database is the ResFind that database, and you just want to see how many of the antimicrobial resistance genes were found in your genome by BLAST. And you will just look for the second column, for example, which is the accession. Of course, you just say $2 here, and then you will get the second column back. But Oak can also be used for filtering your results. So you can say that I just want to keep those lines where $1, 1, 2, 3, 4 divided by $5 is more than 0.5. And then we are just getting back the first line, of course, where this one divided by this one is more than 0.5. So it's more than 50% covered. So this is the main usage of BLAST. There are other things you have to know, especially if you want to use the BLAST P, because if you search for nucleotide sequence, the nucleotide sequence substitution matrix is quite simple. I saw that there are a few sophisticated substitution matrices, but I think the default I hope it will be there. Here, no. I just want to show you the most simple one, which means that everything is 1 or 0. which is not, I don't know if BLAST is working with that or BLAST is working with something like this here. Oh yeah, it's here, the simple model. The simple model says that if you have an A in one sequence and in the same position you have an A, then it worth 1 point. The C with C is 1 point, T with T is 1 point, and G with G is 1 point, and everything else is 0 point. So why you need this, of course, is because the way how, for example, percentage of identity is calculated or the identity score is calculated is by adding up this point. So if 2,940 nucleotides are exactly the same, then in each position you will, or the BLAST will score 1 for all of them. And if there is a SNP, then it will be a 0 in that position, so the score will go down a little bit. And this is the substitution, the simple substitution matrix for nucleotides. But there are actually sometimes a bit more complicated mutations as well. So there is sometimes a bit higher chance for certain mutations in the DNA. But even if we just use the very simple matrix, that's usually enough for nucleotide BLAST. For protein BLAST, and we won't do protein BLAST because it's exactly the same. It's just say BLAST P, but you are in the same way you are using -dB, and you have to give your protein database, and then -query, and you have to give your protein sequence. And everything else is the same. You can do the same kind of -out FMT6, and you will get back the same kind of tabular format as we got back here. The only thing you have to keep in mind, if you do protein BLAST, then the substitution matrix is a very important thing. There are default substitution matrices. I think the BLAST is probably using one of the blow-sum matrices. Let's check if it's there. Sequence option. Yeah, it uses, so you can add other different matrices by minus matrix. If you don't define it, it will normally use the blow-sum 62, which was a matrix created a long time ago based on the comparisons of many different protein sequences. So the way how they did that, they looked for homologous protein sequences, but not completely homologous, quite similar DNA sequences, and they checked what are those amino acid substitutions that are more tolerated by proteins. So if you think about the 20 different amino acids, you can imagine that replacing a hydrophobic residue to a hydrophobic residue is way much more tolerated in a protein than replacing a hydrophobic residue side chain to a highly charged, negatively charged residue side chain. And this is what in this blow-sum matrix is actually defined. So if you look for this specific substitution matrix, then you can see here. Let me just show you an example. So, of course, the diagonal is always a positive number, but even the diagonal is not the same. So if you look at, for example, there are some residues that are so special, so they are not really tolerated to change to anything. So if you have a cysteine, then it usually gives a much higher score if you have a cysteine in the same position, or the histidine, or so on, or the tryptophan. And I think the glycine probably has the smallest, not the smallest, but quite small score. And what I told you, for example, if you look for negatively charged residues, glutamic acid here. If you look for glutamic acid with the aspartic acid, which is this one, then you can see that it's actually a positive number. So changing a negative charge residue to another negative charge residue is not a big change. Or changing a glutamic acid to glutamine is again not a big change, because it's a charge change, but it's not a big change because they have the same size. But if you look, for example, a big hydrophobic side chain, tryptophan, and changing to a negatively charged one, aspartate or glutamate, it's a -3 penalty. So all these scores are adding up, and they will provide your final homology score for the two proteins you are comparing. The biggest problem with this one is that it's working perfectly for globular proteins, but as we know, globular proteins are only about 1/3 of our proteins. In eukaryotes, in prokaryotes, it's a bit more because there aren't too much disordered proteins in prokaryotes. But usually these substitution matrices are working, for example, extremely bad for transmembrane proteins, especially the transmembrane regions of transmembrane proteins. They are behaving very differently from the globular proteins. So this thing has to keep in mind when you are using substitution matrices, but that's all for BLAST. So as you can see, if you use BLAST, you are looking for sequence similarity. There is an alignment step when BLAST is trying to introduce gaps if it thinks that by introducing one insertion or deletion, you will have a much better matching between the two sequences. But the whole search is based on A, complete identity. So if you have the same nucleotide or the same amino acid, that's a very high point. B, in proteins sequences, it is based on the characteristics of the residues. And C, it also based on the introduced gaps or deletions or insertions between the two sequences. You can already think about so if you wanted to find nucleotide homology and the two species are very far from each other in the phylogeny, there is a good chance that they are just drifted away from each other by the nucleotide sequence, even if they are encoding very similar proteins. You can have a huge difference between the two nucleotide sequences. Of course, there is also paralog evolution. We know that domains can evolve multiple times. And if they are evolving multiple times from completely different nucleotide sequences, then there is a very good chance that you will not find the homologous DNA sequence. So if you try to find out, for example, a homologue for a completely unknown gene, so you let's say you are isolating a bacteria, a colony sequencing, you are annotating with PROCA, and then you find that the genes that you are interested in are annotated as unknown function genes. Don't start looking for DNA homologues. Start looking for protein homologues first with BLAST-B, because you will have much higher chance to find something. But even BLAST has its limits, because you know that there are everything is just based on a very general substitution matrix. This is a hugely simplified matrix compared to the complexity of biology. In many cases, domains are not behaving the same way. Even within a domain, there can be a huge difference. If you have a globular domain, so if you think about any kind of three-dimensional structure, inside, in the middle of these domains, you have a hydrophobic core. If you change a hydrophobic residue inside to a charge residue, that will be a very destructive thing for the domain. Let's say that you have a tryptophan or you have a leucine inside and you are changing it to an aspartic acid. It's a -4 penalty. If you do the same thing on the surface, because sometimes you have hydrophobic residues on the surface as well, that maybe doesn't do any difference for the domain. Maybe there won't be any difference for the function of the protein. Exactly the same change, which for the BLAST means exactly the same, -4 penalty, can mean a huge difference biologically. It can destabilize the whole domain, or it cannot, or it may be a completely harmless change. So if you want to look for genes, or especially parts of proteins, domains more specifically, then I think the better way than do BLAST is to use a hidden Markov model based search. And this is our next thing what we will do. The program that you should use is called HMMR, HMMER, or sometimes it is also called Hammer. Luckily, this is also installed. It is not HMMER, this is not present, but there are different HMM programs that are installed that are part of this package. So if you just say HMM and push the tab, then you can see that we have HMM Align and Beard, and I think HMM Search as well. And this is what we will use. So in this case, you are not looking for a sequence in a big database of sequences, but you are using a sequence query, but in a database of domain descriptions. One of the best websites for looking for domain descriptions is a PFAM website. In this case, I already have one PFAM, I think. But if not, we can just, we can just go to the home. It's the pfam.xfam.org. That's the website address. And for example, if we want to look for our MAC-A gene and MAC-A domain here, then you can see that there is a domain identifier or a domain entry for the MAC-A. You can see here the HMM logo, and this is the most important thing here. This is extremely different from BLAST. So what you see here, and I think we already spoke about this earlier, the sequence logo will give you a positional preference for different amino acids. So for example, if you look at here, in this position, the tryptophan, which is the Y, in position 166 is the most common or the most preferable residue. But in the same way, there are positions, probably surface parts of the protein that are absolutely not conserved. So as you can see here, this is not a conserved region. It can be almost anything. It is very far from 100%. So there are some preferences here, but they are very small. So the y-axis here, the y-axis here is giving you a kind of preference for or frequency or number of. And again, what you see here, yes, D and E is right next to each other. D is for aspartic acid and E is for glutamic acid. So in this case, probably the BLAST would work as well in just this position, because these are both negatively charged residues, or here R and K, which is the arginine and lysine, or lysine, right next to each other, which are both positively charged residues. Or here, two hydrophobic residues, but there are so many cases which is when it is not as simple as it is simplified in BLAST. So what you can do, you can download these logos one by one. So if you're just interested in the presence of a special protein product or domain. Of course, you have to translate your DNA sequence if you have DNA sequence, but that's usually not a big problem. You will see that we already have the protein sequences, for example, for our de novo assembly. And you just simply download here at the curation and model. You can download the HMM for this family. You don't have to download this because I already downloaded it. So if you list your data folder, which is the common data folder, you can see that we already have that maca.hmm there. So this is the file which I clicked here. I actually right-clicked and copy link and downloaded the HMM file here. This is a text file, so we can look into the file. If you want, you can also look into /data and mac.a, not mac, a.hm. And what you see here is, this is here the description, the long description, the same thing, what you see on the sequence logo. So just not by graphically, but with text. The big M, which is the methionine, is somewhere here. I think this is maybe about the second position, which is yeah, I think this number here. But it's not important at the moment. The important things are that you also have the description of the domain, and you also have these numbers here. These are one of the most important numbers if you do search with an HMM-based search. It's also here. So these are the numbers what you have in the text file, the gathering cutoff, the trusted cutoff, and the noise cutoff. And from these three, actually the most important is the gathering cutoff. So you will have a number at the end. You are searching your genome, and the program will say, okay, I found something that seems like this domain. And you want that score, which is given, higher than this score. If it's very close, that means that maybe it's not a highly reliable hit. If it's way much higher, then you can be very sure that it is a real hit. You can use, again, the e-value, but e-value is very dependent on your database size. So I usually use the gathering cutoff. And let's do this. So again, anyone can do this, especially if you have your Proca folder. If you don't have your Proca folder, just follow what I'm doing. So if you have your Proca folder from our de novo assembly and annotation before, what we did, if you don't have, you can access actually to my folder. Let me just copy the file from here. I don't know how many of you have this. So if you look at the Proca folder, in default, there are so many different files created. We talked about this before, the GFF file for the gene names and positions. The GBK is the same, but in gene bank format, the table format of the genes that are annotated. But also we have this FAA file. And the FAA file is actually all the genes that were annotated are also translated to protein sequence and put into a multi-FASTA file. So I will just copy this into the data folder so we can all do the HMM search. So if you just look into that, into the file, you can see that this is a very normal FASTA file, one ID, one sequence, one ID, one sequence, and all the listed gene products that were found by the Proca in our Staph aureus genome. We actually know that there is a MAC-A gene in this one. So if we perform a search, then we have to find that gene inside. So the way how you do this is by using HMM search. And if you just say HMM search, then you will get back a very short usage help. So you have to say HMM search and the option, and then the HMM file, and then the sequence you are looking into, the sequence database in this case. But if you want to see a bit more, of course, you can always say -h, and then you will get back much more comprehensive and detailed help. But let's just do with the default without any options. HMM search, the HMM file, and the sequence. So if you are in your own folder, then just say HMM search /data / maca.html. So we are giving the HMM file first, and then /data / proca something FAA. Okay, this is what we are looking for, and this is the whole proteome actually of that Staph aureus genome, what we assembled a few meetings ago. And if you say press enter, this is again extremely fast, and you will get back a nice, again, absolutely useless in terms of programming and parsing, a useless output, but you can at least see what is happening. And when I told you that your score has to be higher than 22, much higher than 22, then we are happy because our score is actually 168. So this is a really, really good hit. There are other things here. I don't think this is extremely important now, how these aligning to your PFAM. This is the most important line, that your domain is there. Also this one here. Again, if you don't really want to check all your HMM searches manually, but you want to do this, you want to parse or digest the output with some kind of programs, for example in R, and you want to have a nice list of your hits. So if, for example, if you are looking this MAC-A in 200 different genomes, that's perfectly okay. You can do this, but you probably want to have a table format output. That's not a problem. So if we go back to the help, there are options on controlling options, directing output. So first, of course, you can put this output into a file, but we don't want to because this is not really usable. But there is a table out, minus table out. I have a flying here. So if we go back to the same command, let's say minus table out. Is it tbl out? Yes, tbl out. And we have to give a file name here. So let's, because we are in our own directory, let's just give mac-a pfam output.txt. Okay. If you do this, something is not good. Yeah, the problem is that you have to give all the options before the files. I forgot this. So if you look at the syntax here, you have to say HMM search, then the options, then the HMM file, and then the sequence. So I put the option at the end, which was a mistake. So I just go back here with my cursor, copy and paste this one from there to there. And it will give you, sorry that it just went away fast, that was the command. We get back the same output to our screen, but we also created a file called mac-a pfam output.txt. if you look into less make a underline pfm output.txt, then you will see that it is much easier to parse actually the program, especially because all the lines that are just blah blah, they are all starting with a hash mark. So you can easily tell your program that ignore every line that is starting with this and only look for those lines that are here. And what you have here is that you have your target name. So this is actually the config identifier, or probably the gene identifier or protein identifier in the FASTA file. And you have here the hit, the gene you were looking for, and also you will have here the score and a lot of other things. In default, I think it doesn't give you the position, so where it is. But in this case, it's the whole entry actually, because the proteins are separated one by one. So in this case, this is not that important. Where is it? So that's the HMM search. Again, if you look for certain domains or genes, then I would suggest to download the individual HMMs from the PFM database. If you just want to see everything, you can also download the whole database, which I don't know why it's not really working, but I think somehow you can copy the FTP link from here and then put into, I think if you just copy the link and put in Finder. So in the Finder, you just have to say go and connect to server, and then paste this PFM link here. If it asks for the connection, you just say you just want to connect as guest, and you are connected. And always go to the current release, and from here there will be quite a lot of files, I think. Here. Don't do this, just look at what I'm doing, because then you will slow down your machine as well, like I did. Okay, while it is coming down, I will tell you the hybrid way I found. Is there any questions for the HMM based search? Or the BLAST? I forgot to ask if there is any questions for the BLAST search. Okay, if not, then we are running forward. And the next would be the, okay, so there was an interruption in the connection. I don't know if, okay, it's here, yeah. So if you finally reach the same place as I reached, then you have to download, if you want to download the whole PFM database, which is thousands or 10,000s of different domains, what you want to download is the PFM-A, and PFM-A HMM.gz, I think I would download this one. So these are the HMM files for all the PFM-A domains. PFM has a P, there is no B anymore. The A are the domains that are highly curated and really reliable domains. I think the C is much more some kind of automatic computational pipeline, which is collecting sequence information. But for you, the HMM files are the most important, and I think you only need this HMM.gz, probably the .gz as well. I think this one is only the HMMs. So if you look into the file, I think this is the HMM file, and probably the .html is also including all the information you can find on the website as well. So it's probably including this summary, description, probably some of these domain organization graphs. So I don't think you need that. If you're really looking for more information on domain, you just have to go up to the website. But if you want to download the whole PFM database, this is what you need, PFM-A .hmm.gz. Of course, it's gzipped, so you have to unzip first if you want to use. Okay, so the next one is the direct sequence search, and for this one, I sent you a link about the eukaryotic linear motif database, which is actually not the best for most of you, because most of you are working on prokaryotic genomes or genes or proteomes. But to understand this, I think it's okay. But I actually also found another one, which is called Prodoric2, which is also a kind of sequence pattern based, but prokaryotic, and it's nucleotide. So the difference, the M is eukaryotic and protein, and this is prokaryotic and nucleotide search. And it is not exactly the same. So this Prodoric is somewhere between the HMM search and the pattern search. I will tell you why. If you just look for one element here, one entry, so if you browse just look for Prodoric2, and this is I don't know, the second one. So what you see here is that it is containing consensus sequences. It's almost the same as an HMM logo, but it's actually not the same. The scoring is very different, and also in HMM, there are also gaps, insertions and deletions are allowed. Here it's not allowed. So what you see here is just only how the different nucleotides are conserved in different places. The thing here is that if you have a G at position 11, I think it's also allowed, because it's the T and the A is not the obligatory residues, I think, here. But it's also depending on the whole sequence context. They are probably not that conserved, but maybe these ones are, yes. I saw someone, oh yeah, here. So as you can see, for example, in the position 10, T is more common than A. This is something what you cannot really do with a sequence pattern search. You just say that, okay, in position 10, I can have T or A, but there is no higher preference for T. In this case, it would get a higher score if you have a T there. So this is a kind of middle way. I don't know if there is any command line program for example, to find these promoter motifs in genomes. There is a way here on this website to use your FASTA files and search for these virtual footprints, but I haven't found any. I mean, maybe this I think this is just downloading this table actually, which is the scoring table for the sequence, but it's not the program. So maybe you have to write your own program if you want to search. The M is way much more simple, and this is our last way of finding sequences today. So if you look in the look at the M database, it will it is containing a lot of different recognition sites. So if you look at the left-hand side, yeah, it is m.eu.org. The CLV means it's a cleavage site. So for example, the cleavage site for caspase 3 and caspase 7 proteins. So what is the protein sequence that is recognized by these proteins? There are degrons and docking sites and ligand sites. Modification sites are very important. Modification sites, for example, phosphorylation, glycosylation sites. So the sequence that are able to glycosylated or phosphorylated, and also there are some targeting sites. But the most important thing here is that your sequence is defined completely differently what we saw until now. So there are these very small characters here, but I will just click on one of them. And this is something we already discussed in like very small detail when we spoke about string recognition in our during the R programming. So this is actually a regular expression definition. So what you see here is that the first amino acid has to be D, S, T, or E. The next one can be anything except P, which is a proline. So this sign in regular expression means that anything else than P. So all the other 19 amino acids. The next position can be anything except these ones. So this is the third position. The fourth position has to be a D, an aspartic acid, and the fifth position has to be something from this fold. So the Elm is only looking for these very short protein sequence pieces. And sometimes this is not extremely well defined. In this case, it's not bad, but even I think this kind of sequence pattern can be quite common. But if you're looking for, okay, if you see something like this, this is a CAN box, very simple. The nucleotide or the amino acid sequence are looking for anything then a lysine and a glutamic acid and asparagine, I think, then, and then anything else. I mean, this is not really extremely well defined, as you can see. So it is very easy to find false positives, of course, and I will speak about this soon. But also, if you look at a bit more complicated one, for example, this one. But again, for this, you just have to understand what the regular expression is. So what is said that in the first position, there has to be an N or Q, which can be there or cannot be there. The 0 and 1 means that this nucleotide or this amino acid, sorry, this amino acid can be there 0 or 1 times. So it's not obligatory. Then you have 2 dots, which means that you have 2 anything, 2 amino acids from anywhere. Then the next position is I, L, M, or V. The next position is S or T. The next position is D, E, or N. The next position is F and Y, and so on. And then, for example, here you have a dot, which means anything again, 2 or 3 times. Minimum of 2 times, maximum of 3 times. This is why this is in a curly bracket. And then a positively charged residue 2 or 3 times. And then anything, but not a negatively charged residue, D or E. So there are so many cases when you find something which is not a true cleavage site for caspase, but it's just by chance matching with this pattern. And this is always the biggest problem with short sequence search. So if you think about promoter patterns, if you think about, okay, actually, if you are looking for restriction enzyme sites, they are very well defined. But just think about think about the restriction digestion. When you are doing restriction digestion, you almost never has a 100% digestion efficiency. Why? First of all, because you have much more DNA. Second, because maybe the DNA is not accessible for the enzyme. And if you think about, for example, here in the protein world, for doing a phosphorylation on a protein, that kinase enzyme, which is doing the phosphorylation, has to access that part on the target protein. So if you look at I think the phosphorylation sites are extremely weakly defined. And this is a kinase site, for example, here. I mean, this is almost, I mean, you have to have two positively charged residues right next to each other, then something else, and then you have a serine or threonine. The reason why they are in brackets, because this will be the residue which will be modified by phosphorylation, and then you will need anything else than a proline and then 2 amino acids. So it's extremely weak definition. You will have false positive hits from this a lot. If you are interested in pKa phosphorylation, and you say, oh, my target protein is phosphorylated by pKa because I found this, that's maybe not true. You have to go after all of these and try to find, for example, if it's on the surface or if it's buried. If it's buried in a domain in the middle, then pKa will not be able to access this sequence. It will not find, it will be completely blind to this sequence part. If we speak about this kind of third option, when we are looking for exact nucleotide or protein sequence, you always have to keep in your mind that if you use a short consensus or a short search, what sometimes you have to, then you can end up with a lot of false positivity. If you use a longer one, for example, if you look at these examples here, you may say, why don't we go further away from this motif and also define these amino acid positions and these amino acid positions here? Because you cannot, because it is so variable. So it just doesn't do anything with your motif actually. So it's not the part of your recognition motif. So, and it's also true for nucleotide. There are many times when you are just looking for a small promoter sequence, and you can find it's the same promoter sequence within the gene as well, because it's just too small. So in a coding sequence part, which is not a real promoter sequence motif. But if you go up and if you define more nucleotides, for example, in the case of nucleotide search, then you have a chance that you will miss it because there is a SNP there. So let's say that you are looking for gene sequences by complete matching. So you don't want to use BLAST, you don't want to use the HMM search, you want to find your gene exactly with the same DNA sequence as you have it in your database. In many cases, you won't find your gene because it is just one SNP different here or one SNP different there. But of course, you don't know where the SNP is. You cannot just allow all the third nucleotides in the codons that are not causing any amino acid change to be like variable, because even amino acid change can happen within genes without destroying the protein function. So you have to be very careful if you look for small nucleotide or protein sequences, and you always have to do something else here. So here, don't look for a pKa. First of all, don't look for a pKa recognition site in a protein which is not phosphorylated, which is never phosphorylated, because probably that won't have any kinase motif on it, or phosphorylation site. So I would suggest you, if you look for these kind of short sequences, only look if you know that your protein is cleaved by another protein, if your protein is phosphorylated by another protein, if your protein is glycosylated then you can go into the details and find the potential or candidate sites. And then you can try to find out which one is the real one by doing experiments or by doing a little bit more bioinformatics. I think I will finish here today. There is no extra program for this one, but you can always just simply copy and paste this from to an R. Let me just show you. And use our, oh, my RStudio, I think, is updating, but it's here now. So let's just do a simple thing here. I will use this modification site. I'm just simply copying the pattern. As you all remember, if you load in the string R package, which we used before, string R package, then you can use your string search. So if you say take str match or detect str match, and then we will put here the string soon, but first I will put here. So this is what you just simply copy here, and the str match will understand it. Even if there are brackets, it will understand. And for here, I will just put in one of the sequences where it can be found. Oh, we don't have any sequence here. No instance. Oh, I was able to find the one where there is no sequence for it. Nice. I will just then copy and paste this one instead. And copy and paste the sequence. Here. So this is the string we are looking for, and this is the pattern we are looking for. And as you can see, it was found, the KRLSMEI, it is down here. And also the phosphorylation site was found. So it is, of course, in the middle. And you can see that this first two amino acids it looks for, it has to be R or K. So this is it. And then there is anything, which is a leucine here. Then there is an S or T, which is a serine here. And then there is anything except a proline. We have a methionine, so it's not a problem. And then two other anything. So if I just simply rewrite this KR to KD, for example, then there won't be any match anymore because the second position is not matching anymore. So if you are looking for sequence patterns, even if it's protein or nucleotide, you can use very easily the R. You know, if you are looking for nucleotide sequence and you know that you have like an A. Of course, if you look for ACG, ACCG, you'll find it. But if you want the second position be a variable, you can, if it can be anything, it can be just a dot. So it can be ACG or T, But if you want to define, if you just want to see A or T there, then you can put it like this. And this will also find, oh, it won't find because it has to be C as well. And then it will be here, the hit. Okay, this is the best way. If you, for example, we did it with Ibrahim when we were looking for specific known mutations within a gene. So if you look for known mutations and you know the surroundings, then the easiest way is to, and you, for example, PCR sequence the gene or somehow extracted the gene sequence, then you just simply put your mutation site in the middle and you define the two. Let's say that this position is the mutation here and you want to extract it. And if you do this, then it will find the pattern and it will also extract the T, which is the nucleotide you are looking for. So it's very easy to extract known mutations from known DNA sequence. Of course, if you want to be more specific, you will put a bit longer flanking 10 nucleotides for both sides of the unknown or the mutation. Okay, I think I will finish here, and I'm happy to have your questions. And yeah, thanks very much, Tom, for your help today.
Yeah, I didn't really do much
in installations, but I hope you follow the searches and I hope you all see that it's so easy, and it's of course way much faster if you have hundreds or thousands of sequences doing it. You can put so if you just think about the HMM search, you can put more than one HMM here, so you can put the whole PFM database, or you can put your selection of domains you are looking for, and you can put as many FOSTA entries here. So if you have multiple files, you can just merge them and use the FOSTA identifier to find out where they were, or you can use whole proteomes or many whole proteomes. So it's very flexible. It's also true for BLAST. You can BLAST a single contig or a single sequence against a single contig, but you can also BLAST multiple contigs against multiple contigs or multiple genomes.